I'm not going to talk about COVID except to say how proud I am to be a member of this university in this extraordinary period. UCSF's leadership is exemplary. Our clinicians, scientists, and students are responding to this disaster, creating diagnostic tests, developing novel therapeutic strategies, designing clinical trials, and tracking outbreaks. We are indeed a powerhouse. The inventiveness and dedication of my UCSF colleagues is outstanding. And against this backdrop, we're proceeding with Pitch Night, the culmination of a class that brings Silicon Valley to UCSF and teaches our community how to create high-impact, scalable, deep science and tech businesses. To quote our industry association bio, innovation will help solve this crisis and nothing will stop innovation. So let's talk about entrepreneurship. We started class on January 26th, a date when the world was still sane. Students expected an intense, exciting exploration of the startup world. Then COVID happened. 4 of the 12 teams you'll hear tonight started thinking about how they could pivot their focus to help with COVID. One student spent last weekend at an MIT-sponsored hackathon to build an app for new ways to deliver care. Another student has sourced a potential serological test in China and is seeking ways to test its efficacy in the US. Last night, I heard from a student who postdocs at the Abadi Lab that he has gotten permission to work on a rapid diagnostic test. Our entrepreneurs are doing what entrepreneurs do, seeing a need and seizing it. Let me tell you about this cohort. There are 4 first-year med students, notable because this class has intense time demands. Med students typically drop out after week 2. These med students not only completed the class, but are among the top performers. There is a team of pediatric surgeons who sometimes can't be in class because they're doing surgery, one of the better excuses I've heard. One student joins team calls from India where he's been stuck. There are PhD students and postdocs, master's students, faculty and staff, people from the business community, Berkeley, Haas, and Stanford. We strive to mix our UCSF community with others for diversity of experience. If a team is not led by a UCSF person, that team lead has to recruit 1 or more UCSF people to join in order to get into Startup 101. Let me tell you about the class. Our goal is to teach how to take a scientific or clinical idea and translate it to a startup. Motivations range from wanting to see what this is all about to absolute commitment to doing a startup. In the words of one postdoc, I'm dead serious. There are 2 business community-led teams who have already launched. Some of the teams you see tonight will proceed on the path to starting a company or have already started one. Others will delay because they're still in school or need to mature the science inside UCSF. And still others aren't serious. They've been exploring entrepreneurship as a possible future path. In addition to creating an executive summary and pitch deck, every team completed 20 customer discovery interviews to help them understand the market. We helped create NIH's I-Course syllabus and are steeped in customer discovery. I want to give our thanks to our many guest lecturers and mentors who helped make this class the top life science on healthcare entrepreneurship class in this country. Teams will be judged tonight by 6 investors, both angels and VCs, who represent a spectrum of approaches and sector interest. I'll let them introduce themselves in a minute. We have 2 valuable prizes for tonight's teams and want to thank the angel groups who are providing them. First, thank you to Life Science Angels and founder chairman Alan May, who is one of the judges tonight. Life Science Angels will give the top 2 teams the opportunity to present to the screening committee, bypassing hundreds of applications and moving to the top of the pile. So thank you, Alan. These teams will also be mentored by an LSA member. And thank you also to Band of Angels and Judge Rich Julis for contributing a mentoring session to the top 2 winners. The band has done this for the past few years, and it's been immensely helpful for the teams. Tonight's format is a 7-minute pitch by the team lead, who will be joined by team members for a 2-minute Q&A by the judges. So now I'd like to turn to the judges and have them introduce themselves. Maybe we'll start with Ken. Ken Haas.
Okay, sorry, just had to unmute while I was listening. So I'm Ken Haas. I'm a partner at Abingworth, which is a life sciences only firm that has raised 15 funds over 40 years. We invest in everything healthcare, mostly therapeutics these days, but also devices, diagnostics, healthcare IT. We have offices in London, Boston, Menlo Park, where I am. We invest in all stages, including public, though at least half our investments are in seed or Series A rounds. I personally have been at Abingworth for 15 years, which has included leading our investment in a number of UCSF startups, a couple founded by Kayvon Shokat, one recently founded by Jim Wells. Prior to Abingworth, I spent 25 years in the management of both early stage and public high tech and biotech companies. Among other things, I was part of the founding management team at one of the first bioinformatics companies, and then was CEO for 10 years of a publicly traded software company. I've been co-chair of the advisory council to the Neuroscience Institute at Stanford as well. And I've been a judge at Pitch Night before, and I'm happy that Stephanie's invited me back.
You're welcome. Okay, how about Alan May? Let's keep this to 60 seconds each, guys. Alan, unmute, please. Yeah.
Figured that out. So good evening. Thanks, Stephanie, for having me, and my congratulations, along with the others, for putting together such a phenomenal program. This is seeing where this has come in the last 7 or 8 years since you've been doing it is really a thrill, and what an impact you've had on the community. So I am a founder of Life Science Angels. We're one of the largest groups in the country, and we're unusual a little bit in that we're sector focused. We only invest in med tech, biotech, genomics, diagnostics, and digital health. And everyone in the group has commercialized technologies in the sectors. So we tend to be vertically focused rather than horizontally focused, as most angel groups are. Because of that, we've centered on early stage investment. We're about 17 years old. We've invested about $70 million. This is angel money. So as you all know, that means it's our own money. We don't manage other people's money or get paid in any way other than the success of the companies we invest in. We cannot make money if the founders of those companies do not make money. And I think never has there been more alignment between early stage investors and founders in that respect. I was a venture capitalist before. I've started 8 or 10 companies myself, and all of them have been in the life sciences. So with that, I'll stop.
Thank you, Alan. Neil, you're next. 60 seconds, please.
Everyone, Neil Tawari here. I'm managing director for our digital health fund at Novartis. Novartis is a publicly traded pharma company with over 50 billion in sales a year. We have 2 different investment arms, one that focuses on biotherapeutics and a second that focuses on digital health. I lead our digital health investing efforts. We typically invest in Series A to B companies, average check size between 2 and 5 million, but we have the ability to write checks as small as 250,000 to as large as 25 million. We have 20 companies in our portfolio. I sit on 5 boards, most of them in the Bay Area, for a variety of companies. And we typically invest across what we call the spectrum of pharma, from drug discovery through development through commercialization and everything in between. And the last piece I'll mention is that outside of investing, my background was in product development. I'm a biomedical engineer by training, launched 10 plus medical devices in my career, and I've spent a lot of time building software applications, and I utilize that across a number of our portfolio companies that we have today.
Thank you, Neil. Okay, Lynn, you look like you're ready.
Yeah. Hi. Thank you, everyone. And thank you, Stephanie and UCSF for having this opportunity. Lynn Chow O'Keefe, founder and managing director of Define Ventures. We're an early stage sector focused fund purely on digital health. We do everything from incubations to Series B investments. We have 9 investments in the fund, and we're really proud of our entrepreneurs and our co-investors that we've invested alongside. People like A.G. Brenstein, who's a former founder of Humedica, is an entrepreneur we've invested in. Tom Pike, the ex-CEO of Quintiles as well. We've co-invested alongside Sequoia, Google Ventures, Bain Cap, Khosla along the way here. So I'm excited about our portfolio. And then before this, I led the digital health investing effort at Kleiner Perkins and was also at Apex Partners investing in enterprise software. So thanks again, and thank you for having me, Stephanie.
Thank you, Lynn. Thanks for coming. Okay, Rich. Rich, you need to unmute.
Okay, thank you, Stephanie. Yeah, I'm Rich Julis, co-chair of the Life Science Committee at the Band of Angels. The Band is one of the earlier angel groups going back for about 20 years, so we've made numerous investments over the years. About a third of what the Band does is life science investing. We try to not only invest, but to advise and mentor companies. Many times we bring companies back from an early stage, then invest later. Early part of my career was with Hoffman LaRoche and Merck, and then subsequently with several Bay Area biotech companies, late stage private and early public companies.
Thank you, Rich. Francisco, you're the last, I believe.
Thanks. So I'm Francisco Jimenez. I'm a principal at 8VC. We are a $640 million, or a current fund of $640 million. It's a generalist tech fund with about 30% allocated towards what we call bio-IT. That encompasses what we feel is everything from traditional therapeutics development through all of the sort of ancillary functions like tools and necessary infrastructure within the biosciences. Our main thesis here is how do principles from building tech companies provide a meaningful advantage in the biosciences. So there's a lot of things where that doesn't provide an advantage, and understanding that difference is really kind of the nuance we focus on here. In terms of my personal background, I've been on the team, basically helped start our bioscience investing practice 4 years ago. Before that, I ran a data science consulting and recruitment firm. And before that was in academia doing research.
Great. Okay, I think I've got all the judges. Let's get Ruben and APO Plus to the stage, please. And while we're transitioning to our first team, I'm going to tell you a little bit about it. So this is a team of first-year med students. Again, they're the ones who've been amazingly dedicated to this. And I've asked all the team leads to tell me something about why they're doing this. So while Ruben, I hope you're getting onto the stage. I'm just going to tell you a bit about Ruben and why he's doing it. So Ruben said, forming this idea into a startup was part of the reason I decided to come to UCSF. I have been working on this concept for the past year and a half, especially while I was interviewing at MD, PhD programs across the country. I was convinced that if I can get this idea to work, I could help a lot of people and open up a new door in the type of medical care we offer patients. There was nowhere that I knew with a better reputation for biotech startups and access to incredible scientific resources than the Bay Area. I didn't want my idea to never reach patients or change medicine. And the only way to guarantee that it reaches patients is to make a startup and do it myself. So I pitched the idea to Kaylee and Ernesto, that's his team, when we all arrived at UCSF. We went to grab beers at Triple Voodoo, I guess that's a good spot, and decided to give it a try. Taking the class has been an eye-opening experience. As students and scientists, we never learned how the business world operates. Class opened up to us all the concepts that really matter when deciding what makes it to market and to patients. Not every good scientific idea is a good product. And that's a critical distinction to recognize when you're talking about new treatments. So that is why Ruben is leading off and in fact has learned what we want him to learn in this class. So Ruben, are you there?
Yes, I am, Stephanie.
Okay, good. And can I see your video? Yeah, I think I just saw you quickly. Okay, and you've got your slides.
Yes, ma'am, I can share them now.
All right, please do, and let's kick it off.
Can y'all see this?
Yeah, we're good.
Perfect. Okay. Hello, everyone. My name is Ruben Hogan. I am the team lead for APO Plus. We are a drug delivery platform, and we believe that we have the technology to deliver cures exactly where you need them. In order to explain to you why we exist, I first have to tell you the story of Victoria Gray, who will go down in history as the first person in the US to receive a CRISPR gene therapy. She needed it because she suffers from a condition known as sickle cell disease. It's caused by a single base pair mutation in the hemoglobin gene, and therefore is an ideal candidate for a simple change. So when doctors cured Victoria, what they had to do was take bone marrow cells out of her, edit them, and then put them back. That is a time-consuming and costly procedure and can be life-threatening. And it really begs the question, what does a better CRISPR gene therapy look like? We think that there are three components. First, it needs to be safe because we're talking about using this in patients. Second, it needs to target those exact same cells that we had to take out of Victoria. And third, it needs to be able to create long-lasting change. We agree that gene therapy is here and it can do a lot of good, but in its current form, it is not scalable and therefore it needs delivery. And to be fair, there are delivery systems that exist, but none of them are suited for the job. The current favorite is something called AAV, a virus. And considering all the news that's going around viruses, we're pretty familiar that they don't always come with good things. In the case of AAV, it can trigger an immune response that can be dangerous for the patient. It also only really targets the liver and so therefore isn't of use to a patient such as Victoria. And finally, it can't really fit all the components necessary for gene editing because of its small cargo size. And as we've learned through our shareholder interviews and also through reading, delivery of CRISPR gene therapies is still a largely unmet need. And if we want to make the benefits of this therapy widely available to those who need it, we need a better delivery system. And that's where APO Plus comes in. We focus on safe, targeted, and effective delivery of gene therapies to the tissues in need. We start with lipoproteins, which are a naturally occurring class of molecules in the body. And because they are a naturally occurring class of molecular shuttle, we know that they're safe. We then add in nanobodies, which are tiny, tiny, tiny antibodies that have the unique ability, because of their size and shape, to bind to a broader array of surfaces than a traditional monoclonal antibody. And so therefore, we can perform targeting in a way that's not been done before. And finally, we hope to incorporate full-length mRNA transcripts that can encode for all the tools necessary to perform gene editing so that we can create an effective, long-lasting change. And how we put our product together is pretty simple. First, we start off with the core structural component of a lipoprotein. In this case, it's ApoA1. To that ApoA1 molecule, we then attach an antibody that targets our cell type of interest, our tissue of interest. This unique ApoA1 antibody construct is what we hope to submit for IP in the future. We then add in the mRNA component that encodes for all the necessary gene editing parts, and then we add in the lipids that make up the lipoprotein. And we're able to combine these three together in such a way that we can produce uniform lipoproteins consistently, and on the order of 10 to the 17th particles per hour. What we're offering, if we can use these therapies across patients with the same condition, is an inexpensive, low risk, and scalable manufacturing procedure. And how APO Plus works itself, we envision to be pretty simple. First, you administer our lipoproteins via IV. Then, once the lipoproteins are in the blood, they travel around until they see the tissue of interest. Once there, the nanobody interacts and binds to that tissue in such a way that we can then transfer that nucleic acid cargo to the tissue in need, creating that long-lasting change. And you probably guessed it already, we're planning on initially targeting sickle cell disease. It's a $5.5 billion market. And 80% of that market is only really dedicated to treatment of symptoms. By collaborating with companies that have existing gene therapies, our goal is to bring cures to a sector of the market that's currently only symptom reduction efforts. And this is what that landscape looks like. In the top left there, you can see companies that are producing drugs right now, and those drugs work in patients, but they're only really treating the symptoms. In the bottom right corner, you can see that there are companies such as Vertex Pharmaceuticals and CRISPR Therapeutics that made victorious therapy, but they require for cells to be taken out of the body. APO Plus hopes to capitalize on that unique top right space where we can bring cures to patients as they are. And what we envision here is a platform technology. Our business model is going to reflect that. Initially, what we want to do is collaborate with companies such as CRISPR Therapeutics and Vertex Pharmaceuticals and enable them to have their scalable cures delivered to patients in need. Afterwards, companies such as CRISPR Therapeutics and Vertex Pharmaceuticals, and even others that are developing gene therapies, have already communicated and made clear that they want to move to other diseases, such as Duchenne's muscular dystrophy, or DMD. And then after that, to other monogenic diseases that affect multiple tissues. And we plan to assist them every step of the way. Our timeline reflects the two things that we think we need to do in the next 18 months. First, we want to demonstrate our in vitro targeted delivery. And then after that, we want to submit for IP based on that unique APOA1 nanobody construct. After that, hopefully we can begin to our in vivo procedure and demonstrate our targeting there, hopefully in mice first, but then ultimately with the goal of moving to non-human primates. We think that initial 18 months is going to set us up on the process to submit IND in the future. And all of this, we believe we have the team to do it. We're composed of 3 MD PhD candidates that have an understanding of how to perform experiments and analyze data and generate hypotheses in a way that can generate a tool. And then we have 2 mentors who have the understanding of how to take a tool from the lab and then ultimately bring it to market. I'm confident to say that this team has the capability to do this. So what we're talking about and what we've explained so far to you is that CRISPR gene therapy is wonderful and can do a lot of good in this world. But the problem is that it needs delivery. APO Plus is focused on safe, targeted, and effective delivery of these gene therapies to the tissues in need. We are APO Plus. I'm Ruben Hogan. We thank you for your time, and we thank you for your attention.
Way to go, Ruben. Very nice. Thank you. Okay, we have 2 minutes for the judges. Can we bring the other team members onto the screen, please? So while we're waiting for the judges to be ready to ask questions, I just want to comment that unfortunately there's no chance for the audience to ask questions. So this will be just for the judges. Okay, who wants to kick it off, judges?
I can start with a quick question. Great presentation. I guess one of the big questions with CRISPR, and we're investors in CRISPR Therapeutics, is a lot of the CRISPR companies feel that they have the ex vivo indications kind of nailed, beta thalassemia, even to some extent sickle cell. If you guys are solving the delivery problem, why go after the ones that they feel they have nailed? Why not go after the harder ones?
Yeah, that's a great question. These companies feel as though they have it nailed because they have a therapy that's effective, that can work. But the reality is that that whole procedure that I explained to you with Victoria, her having to have her bone marrow taken out of her, the editing and the cost of editing that has to happen in vitro, and ultimately the surgery to put those cells back in, all of that, like I said, costs a lot of time and therefore a lot of money. The way we view it is we offer a unique opportunity for you to cut a lot of that cost, cut a lot of that risk, and ultimately just do it in a simple one-time setting. In that way, we think we can uniquely help these companies use their therapies more efficiently. And our goal is to then work with other gene therapy companies that have other similar therapeutics like that. And then to ultimately, at some point, hopefully invest our way into moving into making our own therapies. But initially, our goal is simply to partner with these companies to help them deliver their therapies.
And if I can just jump in, this is Ernesto Rojas. We're also modular. So if you saw that nano body, we can also bring these tools to other diseases that we didn't just talk about. So again, we are a platform and we are modular.
Thanks.
Great. Any other questions from judges or comments? We have time for one more.
Sure, Francisco here. My major question here is around the sort of partnership-focused business model. How do you back into the actual business model here? Is this some kind of royalty-bearing contract that you're aiming to go to? We've generally found in these sort of enabling partnership programs, specifically around delivery, there's kind of a relatively low cap on upside in the company when you're starting to look at these, like, compared to somebody who has an in-house program. So what's the strategy there?
Yeah, that's a great question. I'll answer it really quickly. So our goal is to initially work with these companies because we want to focus on making sure that our product works very well, very robustly, and has the potential to be used in other settings as well. That's our initial focus. We don't have to want to worry about whether or not that gene therapy can work by itself. But again, our goal is after that initial setting of working with these companies until we feel that our product works well enough that we can then invest and potentially take on further risk in developing our own CRISPR gene therapies, that's when we'll then make that move. But our goal is to ultimately move there, yes.
Great.
Does that answer your question?
Got you, we're out of time. All right, thank you, Able Plus team. And on to Avid and Lisa Kirkamo, team lead. So Lisa, come onto the screen, please, and share your slides. And while she's doing this, I'm going to just tell you a little bit about the team and where they're from. So Lisa is in the Jim Wells lab, a very well-known lab at UCSF and an entrepreneurial one. And she said, we're a group of 5 scientists with a growing passion for entrepreneurship. Taking this class has been instrumental in improving our ability to communicate more effectively, recognize market opportunity, and generally think about science with a new lens. So Lisa, please take it off.
Sure, thank you for the introduction. Hi everyone, my name is Lisa Kirkamo and I'm the team lead for Avid Therapeutics. I'd like to start out the evening with a scenario. Imagine there's a criminal in the Bay Area and they're running around, wreaking havoc, doing all kinds of damage. The police can't seem to find him, knows nothing about him. And instead of continuing on with their investigation to potentially find this person, the police decide, let's just round everyone up in the Bay Area. Let's throw them all in jail. This is the strategy we currently take in treating autoimmune patients in the clinic today. Autoimmune disorder is classically one of the B cell, which is a subset of our immune system. And normally, B cells secrete antibodies that target viruses and bacteria for destruction. But once in a while, you'll get a rogue B cell. And that B cell decides, yeah, I don't really want to go after viruses and I don't want to go after bacteria. I want to target cells within my own body. And this leads to drastic consequences for patients with these diseases. Inflammation, tissue damage, you name it. And what we've decided is the best course of action for these patients, just eliminate all their B cells. And this would be a viable strategy, except now these patients are extremely susceptible to both viral and bacterial infection. At Avid, we're developing a therapy that only eliminates the bad guy and lets the good guys keep fighting the good fight. Our initial proof of concept disease is an orphan designated autoimmune disorder called myasthenia gravis, in which a subset of B cells target the musculature necessary for proper breathing and motor function. In 2020, the market for myasthenia gravis was $700 million, and it's projected to increase 10% year after year. But to talk about our therapy, we really have to understand a little bit more about the disease. And so in healthy individuals, the way that our muscles activate and fire is through a neurotransmitter called acetylcholine. And when acetylcholine binds the acetylcholine receptor, it leads to muscle activation. Now, unfortunately, for patients with myasthenia gravis, they develop autoreactive B cells, and these B cells secrete antibodies that bind to the acetylcholine receptor, thus blocking acetylcholine from binding and halting proper muscle function. And as we spoke about earlier, the current state-of-the-art therapies available to these patients remove all the B cells. And while this restores proper muscle function, it does so at the expense of the patient's immune system. So at Avid, we're developing a therapy that removes only the autoreactive B cells, thus restoring proper muscle function while retaining the rest of a healthy immune system. And so you might be wondering, okay, how do they plan on doing this exactly? And to get into that, we really have to understand more about the maturation stages of B cells. And so later in the maturation of B cells is actually when they begin to secrete the antibodies that lead to disease progression. But importantly for us, earlier on in B cell development, those same antibodies are still attached to the cells themselves. So we can leverage this as a selective marker by which to now tag these cells for elimination. If any of you guys are familiar with the use of bispecific T cell engagers in cancer, our strategy hinges on very similar concepts. That is, one end of our biologic binds the autoreactive B cell, and then the other end of the biologic recruits immune cells. Now, once we've recruited the immune cell, we can just let them do what they do best, and that is eliminating harmful and ineffective cells from the body. And in the case of a myasthenia gravis patient, this means halting disease progression and restoring proper muscle function. And so as we spoke about earlier, in today's market, myasthenia gravis is about $700 million. And importantly for us, the predominating therapy within this market is a biologic. And this is good for us in terms of market traction because we're also pursuing a biologic-based therapy. On the right, you can see all of the available treatments, although effective, lack selectivity. And so they all have broad cytotoxic and immunosuppressive effects. And so at Avid, we aim to maintain the effectiveness while gaining on the arm of selectivity. In terms of our milestones, the next 0 to 12 months, we'll really be honing in on our in vitro candidate optimization. So this is biological design and expression, binding optimization, and then proof of principle in vitro that we can selectively eliminate these B cells. From the 12-month mark onward, we'll be shifting into an in vivo phase. And so this is mouse efficacy and PK studies. In terms of what it's going to take, our in vitro phase, we've projected about $800,000. And for the in vivo phase, we've projected about $600,000. So totaling over the next 18 months, we're estimating about 1.5 million to get off the ground. So why do we think we can do this? Well, we've amassed a fabulous team of scientists. Between the 5 of us, we have over 30 years of experience in protein engineering, immunology, and business. And we're joined by an even more fantastic group of advisors. Each of these individuals has experience in starting and running successful businesses. And so in conclusion, we at Avid are really trying to address a vital unmet need for those patients with myasthenia gravis. Current therapies, while effective, all hamper global immune function. So at Avid, we're developing a novel biologic therapy for the selective removal of autoreactive B cells to mitigate disease while retaining a functioning immune system. And what I want to leave you with is the idea that for us, it doesn't end with myasthenia gravis. There's a whole host of B-cell-driven autoimmune disorders that would be amenable to our platform. And in doing so, we can help improve the lives of hundreds of thousands of patients worldwide with these deadly diseases. Thank you, and I'll take any questions.
Thank you, Lisa. Judges, we have 2 minutes.
Yeah, quick question from me. I mean, obviously in these patients, as with most autoimmune patients, safety is key, but it's even better if you can use the better safety to actually then up the dose. Is that also a possibility here, or is it really just about safety for these patients?
I would imagine that dose escalation could be done. That's true. I think right now, what is the limitation is that these are just broadly so cytotoxic and so immunosuppressive that you're kind of having to balance effectiveness and safety. And so with this, because the safety arm is kind of dealt with, you could imagine that you might be able to up the concentration. Yeah.
Got it.
Other questions? Nothing else?
I was wondering why you chose MG as opposed to maybe a bigger autoimmune disease indication.
Yeah. So for us, in the way that our technology works, we really need to focus on autoimmune disorders that are pretty epitope restricted. So these are B cells that are directed towards one antigen and one antigen only. In terms of that, there are obviously 10 to 15 diseases that fall under that category. But for us, myasthenia gravis had the most clearly defined clinical endpoints. It's the most well understood of those autoimmune diseases.
Great, thank you, Lisa and team.
Thanks.
Okay, next, Mike Wang, Clinical Voice. Mike, come up to stage, please. So here's what Mike said when I asked him why did he take this class, and it's pretty compelling. I am a dermatologist and dermatopathologist, that's a tough word, who's been studying machine learning. I am driven to help patients and my colleagues through technology, but the business side of how to bring ideas to practice has previously been a black box for me. Through class, I am now armed with the knowledge and confidence to pursue my goals. The connections that you helped me build during this course are incredibly valuable. I'm glad the course is doing what it's supposed to do for you, Mike, and for the team. So Mike, please take it off.
Hi, can you hear me?
Yes.
Awesome. Should I start?
Start.
Awesome. Thank you for joining us this evening. I'm Michael Wang, and I'm the team lead for Clinical Voice. Clinical trials, its process along with its problem has been brought under the public eye more than ever because of the COVID-19 pandemic. And Clinical Voice is here to help by bringing clinical trials to patients. Clinical trials lack a seamless way of communication. And like a game of telephone, the message often becomes jumbled after only a few relays. While it's fun in a game, it can be dangerous and costly in medical research. From our customer discovery interviews, we hear that currently getting more data is taxing on the patients and clinical sites. And data capture needs to be easy for the patients and occur in real time. The reason that we're hearing that is because we're currently using paper charts as well as human scribes. This has been going on for 180 years. And while it does have its advantages, it's really tedious, it's error prone, and also it limits the granularity and breadth of what it can collect. Paper forms can cause thousands of errors because each clinical trial has millions of data points, and the human transcribes can spend about an hour per patient visit with hundreds to thousands of visits in a clinical trial. So Clinical Voice combines both in one seamless process, delivering the data to investigators, reducing errors and human labor. There are problems in clinical trial at all layers. For each day that product launch is delayed, millions of dollars is lost. And unfortunately, this happens way too frequently. Hospital and clinics also require millions of dollars to maintain for administrative and facility fees. Patients are also very expensive in clinical trial, and unfortunately, 30% of the patients drop out of clinical trials. Clinical Voice offers a remote and voice solution. What it does is to have the patient speak into a voice interface, and natural language processing takes that into structured data before it automatically entering into the data collection systems. So we can be standalone and plug and play with the existing solutions. So it's easy to use. It can be remotely accessed anywhere, anytime. And for anyone who's in pajamas right now, you will be able to use this app. Clinical Voice helps to engage more and diverse patients, therefore reduced possibility of product delays. And we achieve that by reducing the time and geographic barriers patients face. And we offer savings because automation reduces research personnel and the remote access reduces facility costs. The clinical trial market globally is $22 billion, and the US market is $7 billion. The phase 4 clinical trial market within that is $2 billion, and we intend to focus on that first because there's less regulations and increased need for real-world data and evidence. And real-world data and evidence in phase 4 clinical trial are used for medication safety, efficacy assessment, insurance coverage, as well as new indications and label expansion. Clinical Voice competes on being scalable and data-centric with a remote and voice platform. We'll be targeting pharmaceutical sponsors with a monthly subscription fee based on their volume and usage, and they'll be paying a small fraction of the $3,000 cost per patient visit. And they're incentivized to pay us because we offer increased data accuracy and patient retention with decreased costs and delays. We'll initially pilot with CROs, followed by pharmaceutical sponsors with their internal validation process. And followed by that, we'll engage in additional customers in pharma and CRO companies. We'll also drive patient and clinic use through our partnership with our customers in pharma and CRO. Initially, we'll develop a alpha software to showcase our capabilities, and at the same time, we'll sign contracts with piloting sites. Once that's completed, we'll have the beta software for real-world testing, and with that, we'll collect metrics, including data validation, user engagement, cost savings, and we will use that to contact CRO customers, followed by pharma customers. Our team is well-rounded with clinicians, business talent, as well as machine learning experts. We're guided by mentors in pharma, technology, and business. And in summary, Clinical Voice is here to bring the clinic to the patients through a remote platform. We're scalable and data-centric with an easy-to-use voice platform that gives cost savings in a large market. And we initially target phase 4 clinical trials and for clinical voice. Thank you very much for listening. And I'd like to thank again for Stephanie, all our mentors, as well as lecturers in the class for their teachings. Thank you for your time.
Michael. Thanks. That's great. Okay, judges.
Thanks,
Questions for
Andrew.
Mike?
Hi, Mike.
This is, go ahead, Lynn. You want to start?
Sure, I'll do, and then you're next here.
I think we both have a number of investments in this space, so I think
awesome.
Exactly.
to talk.
So we'll first say that we, and Neil probably will say the same, we've invested in a full stack CRO, and we very much believe in decentralized trials for pharma. So I think you're really innovating in an area that will be very big, and obviously in the COVID crisis, we see that today. Yeah. You know, Mike, it sounds like a lot of what the data you are inputting is more patient-reported outcomes.
Yes.
Do you believe you have to expand your platform to include more biometrics and other endpoints for trials, and how would you think about doing that?
Yeah, so our initial starting point is to use voice to collect patient data and questionnaire side effect, quality of life, for example. But in the future, we do believe in expanding that data collection to every aspect of the clinical trial. For example, where did the patient answer those questions? We can also connect to the remote devices to get their heart rate, temperature, stuff like that. And we can also expand to kind of televisit style of linking the physicians to their patients as far as getting physical exams, whatever that can be done remotely.
Neil.
Yeah, and well, great answer. And just building on kind of Lynn's question, my initial reaction is this feels a little bit more like a feature than a full company from the perspective of when you look at other phase 4 focused companies like Medical and those types of companies. They offer the ability to execute phase 4 trials to a certain extent full stack from the perspective of not just collecting ePros, but also by doing consent and managing the trial itself. And there's a significant value proposition there, right? And so when you look at your feature and that what you've created right now, do you think the value proposition is really big enough in terms of the value that you provide?
So let's not take that as something you need to answer right now, but a great comment, something to think about. That's feedback from Neil, who's experienced in this area. Okay, all right, we need to move on. So thank you, Michael. And let me bring on Kyuho Han for Medic. So while Kyuho is coming to join us, his background is he's a Stanford geneticist and a postdoc, and he just had a cover of Nature published. And on top of that, he's been accepted into a top accelerator. So he's going to make this startup happen. This is what Kyuho had to say about class. He said, there may be many good entrepreneurship classes and programs in the Bay Area, but I am confident to say UCSF Startup 101 is the best of all programs for those who are serious about starting biotech and healthcare related startups. This class has been truly helpful with a tremendous amount of information and practical resources, ranging from the basics of startups to high-level business models and strategies. And I swear I didn't write this for him, but he said something that I find really important. So Kyuho, please go ahead. Are you on, Kyuho?
Can you guys hear me?
Yes, now we can.
Okay, good. Okay, thank you for introduction. And can you guys see the screen?
Yes, we can see it.
Good, right. So hi everyone, I am Medic's team lead, Kyuho Han. And to start, I'd like to draw your attention to these numbers. So 93% of cancer drugs fail in clinical trials. And this high failure rate tremendously increased the developmental cost of cancer drugs and the average cost of cancer drugs per patient up to $258 per year, which is actually 4 times average household income in USA. So the bottom line is the system is not sustainable. So why do most cancer drugs fail? They fail because tumors are extremely heterogeneous. Not only is there huge inter-patient heterogeneity, but there's also enormous intra-tumor heterogeneity because tumors are constantly evolving even in the same patient. And this is why cancer is called the emperor of all maladies. So consequently, every cancer patient responds to a cancer drug differently. This plot shows typical responses of individual tumors to a drug, where the drug failed to reduce the tumor size in most patients. And this is a clinical failure of the drug. But what if we could predict who's going to respond to the drug even before we enter clinical trials? So if we can find biomarkers, which are genetic variations to identify such drug responders, drug approval rates can jump from 7% to 65%. To identify such biomarkers, we need to test the drug against the best number of tumor variations in order to reliably measure which variations are consistently enriched in drug responders, which are potential biomarkers. Unfortunately, the pharmas test their drugs only in a tiny subset of possible variations, and consequently, they are severely underpowered to predict biomarkers. It is just technically challenging for pharmas to establish many tumor variations and to track which variations affect drug responses. So how can we tackle this issue? Medic can help pharmas to test their cancer drugs in 2 million tumor samples. We can make this possible by using a drug test bed based on combination of CRISPR and cancer organoid. The first key element is CRISPR, the ultimate tool to edit cancer genomes. There are about 20,000 genes in cancer genome, and with CRISPR, we can simultaneously edit all these genes in one tumor sample to generate a pool of 20,000 tumor variations. Most importantly, CRISPR allows us to easily track all these variations to measure their effects on drug response. The second key element is 3D cancer organoid, which is the most tumor-like cancer culture method. The data from 3D cancer organoid is much more translatable to actual patient than 2D cancer culture model or even animal model. Only downside is that it's quite tricky to handle and scale up 3D organoid for drug test. So the question is it possible to combine these two elements to create the best of both worlds? MEDIC is the first ever and the only team so far to have performed a genome scale CRISPR editing in 3D cancer organoid. We published this in Nature actually a week ago, and I'm the lead and co-corresponding author of this paper. And Stanford University also filed a provisional patent application for the platform described in the paper. And in this work, we show that the single screen in our platform is much more superior to 517 screens combined, performed in traditional 2D cancer models to identify key cancer variations. Importantly, we also identified carboxypeptidase D as a novel drug target for lung cancer and as a potential biomarker for KRAS inhibitor drug, providing strong evidence that we can use the platform to identify biomarkers and even novel drug targets. So in MEDIC, we basically want to scale up this system by 100 times to build an ultimate drug test bed. Here, we will start with 100 different cancer organoids and use CRISPR to create 20,000 variations per organoid, which will produce 2 million tumor variations for drug test. Importantly, we can generate 2 million data points for interactions between a drug and all these variations, which will enable identification of biomarkers and possibly novel drug targets. And here's how the actual system will work. We can get cancer drugs from pharmas and test them on 2 million tumor samples, which will identify biomarkers for targetable cancer patients. Then based on those results, we can help design optimized smart clinical trials, which will maximize drug approval rates at the end. Our target market will be small startups and big pharma with preclinical cancer drugs. And we will achieve two revenue streams here, one from our service and the other from IP licensing and partnership for novel drug targets we can identify. Our total available market is oncology CRO that has 18.6 billion market size, and our SAM is preclinical CRO with 4.1 billion market size. And we hope to obtain 100 million market share here by achieving 500 cancer drug tests per year and by establishing extensive IP licensing and partnerships on novel drug targets. So in the next 2 years, we want to start with a small-scale prototype to validate our platform and expand it to 2 million variations in multiple cancer types. We hope to establish initial partnerships with pharmas at the same time. And we want to achieve these goals with our great team of medics fighting against cancer. We have experts from various areas, including science, medicine, and business. And we have been supported by a scientific advisor from Stanford University, our team mentors, Uder and Brian, and also floating mentors, Golden and Erica. And most of all, I'd like to thank Stephanie for all the teaching and support she has provided to us. So with that, I hope you find our company attractive and join to support us. Thank you for listening.
Thank you, Gio. So let's bring the judges in and see what questions we have.
I'm sorry to start again, but I'll start again. The science is actually really interesting. I'm just a little confused about the business model. As you know, we VCs like drug companies better than we like service companies. How quickly do you think you're going to transition to being a drug company? Because the money you're looking to raise doesn't seem to support that.
Yeah, I think it's a really great question. I think that we will initially start as a platform company, just because as an early startup, it might not be easy to do like therapeutic company immediately, but that's a kind of segue to the therapeutic company eventually. And I think that we can build our platform within maybe like 1.5 years. And then as we are actually like validating our platform, at the same time, we can identify a new drug target. And then if they look interesting, we can actually transition to therapeutic companies. And that is definitely our strategy in the long run.
Got
One
it.
more
Thank you.
question. Sorry, Ken, go ahead.
That's good. Thank you.
Another question?
Yeah, Kiro, thanks. This is really cool stuff. You know, my question, just because of unfamiliarity with these models, is are you only limited to cytotoxic therapeutics, or can you actually do screens for IO type molecules and interesting like checkpoint combinations or whatnot?
Yeah, I think those are like great questions as well. I think that for at the moment, yeah, we can test only cytotoxic drugs because we are measuring gross phenotypes of cancer in our platform. But like we can maybe use other like reporter system that can actually tie to any other phenotypes, other than like gross phenotypes. So this can be definitely expandable to other like phenotypes. But yeah, for the moment, I think we are focusing on the cytotoxic drugs, which are cancer drugs.
Great. Okay, we need to move on. Thank you very much, Kyoho. And the next team up is Image Now, Bruno Larita. So Bruno, while you are getting to stage, let me just tell you a bit about him and his background. So Bruno is one of our industry-led teams. His background is sales and marketing in vitro diagnostics, including 8 years at Bio-Rad. And he told me he wanted to make a real difference through a startup. So when I asked what was it like to be in class, he said, the experience was a bit of a roller coaster. The lows, starting with an idea and having to pivot because so many had this idea before you. And then switching to Zoom from the live class. The highs, finding a pattern through interviews and discovering an unsolved problem that can change people's life and develop into a business. And meeting so many amazing people in the class, mentors, entrepreneurs, and potential customers, all focused on one goal, helping people live a better life. So Bruno, please tell us what you're all about here. Can't hear you, you need to unmute. And just while he's doing that, Bruno's recruited a team of 3 UCSF clinical people, which he was very pleased to find, and it's an awesome team. Okay, you're unmuted, you should be able to speak.
Okay, can you hear me now?
Yes, but I can't see your slides yet. Are you sharing the screen?
Need to share screen. Share. And
good, we got it.
It's not my first slide, but I will start now. And good evening, everyone. Thank you for attending this event. My name is Bruno Larita, and we are Image Now. So this is Marie, my cousin, and her 2 sons. Marie lost her life to breast cancer when she was 43. And this picture was taken when she was undergoing chemotherapy that unfortunately did not cure her. So she was initially misdiagnosed. And by the time she was diagnosed, it was too late. So in the US, 1 in 8 women will develop invasive breast cancer in their lifetime. And that's roughly the equivalent of the population of Florida. So mammography is key to the diagnosis and the detection of breast cancer, but it's not 100% accurate. Out of 40 million mammograms performed each year, 50,000 women will be told that they have a negative result as they have breast cancer, and about 4 million women will be told that they have a positive result and they are actually healthy. So the combined total cost of false negative result and false positive result is estimated to be over $7 billion a year. And the patients are not the only one affected. the family and friends also go through a tremendous amount of stress. So why is that? Well, one is the technology. A mammogram is a low-dose X-ray that allows a radiologist to look for changes in the breast tissues, but it's very subjective. It's not easy to interpret. MRI could do a much better job, but it's also like 10 to 20 times more expensive. And reason number two is the lack of comparison images. As I mentioned earlier, if you don't have a baseline, if you don't have those images, it's very difficult to call a positive or a negative. So this is how medical records are transferred from one center to the other in the US today by fax. And medical images are burnt onto a CD and given to patients or mailed to them. And this is what the radiologist that we interviewed said. One of the quotes that we see here is from a radiologist from Brigham and Women's Hospital. It highlights the frustration and the pain points that all the interviewees expressed. The CDs are crashing, etc. And so in the middle, this would be really helpful, is about ImageNow solution. So what is the solution? ImageNow is a universal, secure, cloud-based platform that allows hospitals, clinics, and radiology group to access a patient's medical images. ImageNow reduces the false positive rate by allowing a radiologist to access all previous comparative images from mammograms from anywhere and at any time. Artificial intelligence can help the early detection of breast cancer. So ImageNow is using its repository of images to implement and set up an artificial intelligence solution. So by implementing deep learning artificial intelligence trained with de-identified images in combination with our network of expert radiologists, we can also reduce the number of false negative results. So by cutting false positive results by 50% and false negative results by 25%, we estimate that ImageNow could basically save about $3 billion a year and many lives. Out of $410 billion spent on medical imaging, 10 are spent on mammography. So with 40 million mammograms a year, if we charge less than burning and mailing a CD to a hospital sending an image to another hospital, so let's say, for example, $3 per image, ImageNow could generate up to $120 million a year. And so for market adoption, we will use the viral acquisition Dropbox model. So how does it work? Basically, we will incentivize our early adopters to register new customers when they receive a request to send or receive an image. In addition, we will partner with health insurance companies and patient advocacy group to further promote our solution. So we studied the landscape across, the competition landscape across 7 axes. And what we found is that competitors focus on some, but not all of these. And so we have an opportunity to develop and present a solution, a hybridized solution that checks all the boxes. In addition, ImageNow will be easier to deploy and more cost-effective. And this will facilitate the penetration in hospitals and imaging centers. So what do we need? Well, we divided the development in 2 phases. So milestone number 1, we need $150,000 and 6 months to develop the minimum viable product, which is the mammography image transfer and web viewer. And milestone number 2, we need an additional $200,000 to develop the AI application. I'd like now to introduce the ImageNow team. ImageNow has assembled an amazing team of experts, medical experts and technology experts. So we have I'd like to introduce Sarah. Sarah is a UCSF and Stanford trained neurologist, and Shadi is a Yale, Harvard, and UCSF cardiologist, and Tess is a incoming UCSF ophthalmology resident, and finally Kenneth is a technology professional from Harvard, and I'm myself a 25 years medical diagnostics veteran. Finally, I'd like to thank all of our mentors, Wan Young, Robert Dickinson, Gordon Murtaugh, for their time and their wisdom. And last but not least, I'd like to thank Stephanie Maris for her mentorship and for creating and supporting this program. In conclusion, ImageNow is a cloud-based platform with AI capability that reduces false positive results and false negative results, and ultimately reduce cost of imaging and improves life. I'd like to thank you very much for attending this presentation.
Thank you, Bruno. Judges?
Hey, Bruno. Okay, one clarifying question. So it's effectively a cloud PACS with MammoCAD layered on top to a viewing system?
Yes, so the first phase is one to allow the image exchange. So in between networks that don't communicate with each other. So for example, if a patient had her first mammography in San Francisco and then moves to Florida, for example, if she doesn't bring her CD, or even if she brings her CDs, most of the time they crash, then she'll be able to access, or the new mammography center will be able to access those prior comparative images.
Right, so the standard is DICOM, presumably, and it's a picture archiving communication system with interoperability across those?
Yes, and especially we want to be vendor neutral as well, which also allow, no matter what the format of the images are, they will be able to be viewed by the web viewer that we will develop.
And this is Neil. Sorry, were you done, Francisco? Yep. We've looked extensively at the space, and I think a couple other competitors you might be missing on your map. Nucleus Health, Microsoft has a solution. Braid.ai is another one recently funded by Lux that seems to kind of overlap with what you're doing. The specific question is a very competitive marketplace in terms of getting adoption by healthcare systems. What is it that's unique about you that'll drive that adoption on the viewer side and on the data side faster than the others who have a head start from you?
So we believe that the adoption has not been very strong, because in all the interviews that we had, none of the imaging centers had such tools except Yale University. So we believe that things are changing now. One is the fact that we are not using fee per service anymore. So by having another type of reimbursement, people tend to adopt those kind of solutions where you have to do less and less imaging. So I think it was a question of timing. I think we arrived at the right time to propose those kind of solutions.
Okay, we need to stop here. So thank you, Bruno. And let's bring Pam England and Nur1 to the stage. And while she's doing that, I have to say Bruno was so dedicated to taking this class. He was always the first one in the room. Sorry about the Zooms, but I could just count on him being there, and he asked great questions. So thank you for your participation, Bruno.
You're welcome.
And, okay, is Pam out there? So while she's finding her way onto the stage, Pam has been UCSF faculty for the past 20 years in the pharmaceutical chemistry department, and Nur1, which she's going to tell you about today, is her passion. So Pam, please get started.
Hi, thanks, Stephanie. Can you see my screen and hear me?
I can hear you, I can see you, and I don't see your screen yet.
All right. I'm sharing my screen, so I don't know what's happening. Video, share screen.
Share it 2 times. There's one share at the bottom, and then again, there's another share option. And you have to share one more time.
Okay, if we can't do this, let's, Pam, let's defer you to
No.
a little bit later.
Can you see the screen now?
No. Okay, we're going to come back to you because we're short of time here. So let me ask Matthias to come to the stage, please. And maybe one of my team can work with Pam and either getting her slides from our backup pile or getting her to find her screen. So Matthias, Medic Health. So Matthias is in the MTM program. For those of you who don't know what that is, it's a master's in translational management, and it's joint between UCSF and Berkeley. His team is impressive. They've really worked well as a team. They've pivoted their idea and they're jumping onto the COVID telehealth opportunity. So by the way, I just heard today that the utilization of telehealth at UCSF has jumped from 2.5% to 55% since COVID started. So you're right, it's an opportunity. So tell us about it, Matthias, and unmute yourself. Great.
Hi everyone, let me quickly share my PowerPoint.
And while he's doing that, he said, I've been surprised by how a team from different backgrounds can have such valuable insights. Especially as a physician, you're taught to be very focused on a problem, which is why it is so helpful to have different backgrounds and opinions around you to open up your view on what is possible. I think it made me much more understanding of different views. So Matthias, show us what you've learned.
Hi everyone, so I'm Matthias, and we are Medic Health. We are a telemedicine company focused on physical therapy. Now let me tell you about Amanda, whom I talked to very recently. So she's a very active person. She loves trail running and she loves all kinds of sports. And about like a month ago, she tore her ACL, had to go to physical therapy, got exercises on a sheet of paper, and then went to surgery. And afterwards, the shelter at home happened. A lot of physical therapy clinics all over the US closed down. And we still have patients like her that need to see a physical therapist, or they might have a lifelong effect on their knees. And so physical therapists at this point are also sitting at home trying to find patients, but the system is not working. So you have both of these groups sitting at home separately and no real way to connect them. What is very interesting though is that COVID-19, as Stephanie said, is forcing everyone to move to telehealth. So even people who never treated patients for physical therapy over telehealth have to learn that now. And so what we want to build is a telehealth physical therapy platform that connects patients and PTs and facilitate scheduling and billing, making it easy, making it very quick to get an appointment, to get reimbursed as a PT. Even though you have your own clinic, you can see a couple of patients on the side. And our long-term goal is not only to provide that interaction once a week with a physical therapy patient, but we want to create, using artificial intelligence and computer vision, a solution that works on smartphones that can track movements and can help people to do their exercises at home, even when they're not seeing a physical therapist. That was our original idea, but that's now moved to the future. So in the past 3 weeks, we have been building a website, and Stephen and Peter have been very good about this. So we could test this out with patients and physical therapists and ask their opinion. And then last weekend, Stephen at the MIT hackathon dove into the depth of what should really the function be of these tools. And he has a background in digital health and telemedicine and compared several potential competitors more from the medical field to look what could this look like. And in the last 2 weeks, we've also been working with Nick outside of this class to create a high fidelity wireframe that we plan to implement in the coming weeks to be able to really start testing this with patients and PTs. So the business model is a two-sided business model, very similar to Airbnb, for example. What is very interesting though, is that the average revenue that a patient gives to a PT per hour is around $125, and a PT usually only sees $40 of that money. All the rest is overhead that goes to the clinic. Now, what we want to do, we want to provide that virtual clinic and take a smaller fee, around 36%, for example. and so we could still maintain a healthy margin and do the reimbursement and make it very easy for the PTs. So the classical customer acquisition cost in this field is around $70 per patient with an LTV around 270. Now, this is the classical physical therapy space, and we have yet to see how this changes in the upcoming digital health space. So the market size is $36 billion outpatient PT in the US. Orthopedics is half of that, and we are planning to target athletes, people who do sports, who want to see a PT once a week for their exercises and for their recovery. And those people, through our interviews, we found were most interested in adopting these technologies. But as I said, everybody will be forced to adapt these technologies, so everybody will learn to use them in the coming months. So the big competitors in the field are companies like Doxy.me and MedBridge that are really built into a physical PT clinic or Doxy to see patients that you're already seeing in your clinic, but there is no real way to get new patients. On the other hand, you have Luna, which is an on-demand physical therapy platform where you can find a PT to come to your home, but there the interaction is really physical. So we want to really combine both to find new patients and to also treat the patients online. The reason we're doing now is because the regulations are changing very rapidly because of COVID. They're less stringent. PTs can use non-HIPAA compliant tools until COVID ends to see their patients, so we can rapidly scale with an MVP instead of having to build everything out HIPAA compliant before we can start. Then reimbursement has been changing. They allow tele-rehabilitation to be reimbursed now, which is very interesting because that's the reason there are few competitors in this space. On the other hand, our long-term goal was creating this automated or assistive exercises at home from a smartphone device. And if we build this platform first, we don't need FDA approval because it will be physical therapists that provide the guidance and the recovery. So our roadmap really is to focus on launching that MVP, getting that customer feedback, and by Q3, be able to create a platform and start acquiring a partner from insurance companies, for example, to send patients our way when they don't know what to do with them. Then as COVID-19 subsides and we're all returning normal to lives and everybody can see a PT without risk of getting infected again, then we plan to start to transition to the computer vision R&D and afterwards really validating that we improve clinical outcomes with our technology. So this is the amazing team. Some of us have a background in digital health and telemedicine. Others are at the moment working on diagnostics at UCSF to try to help the COVID-19 crisis. And we have people with mobility expertise and a lot of physical therapy expertise on the team as well. We have had amazing advisors. Elise and Stephanie throughout this course have been giving great advice. And then Dan Burnett is also somebody who has advised me on more the medical and the regulatory side of things. He has a lot of experience in that field. Then in conclusion, so there is an unprecedented opportunity in telehealth, as we all know, but the space of physical therapy is relatively new, and that's why we want to dive right into it. We're building a double-sided marketplace, and long-term, we plan to transition to these automated home exercises that patients can do on their own time. This is Netic Health. Thank you very much.
Thank you, Matthias. And I misspoke. MTM is Masters in Translational Medicine. I said it wrong. Yeah, it's a great program. Okay, judges.
I like the concept. The question early on is, how are you going to get to the physical therapists? Even approved principal. Do you have those connections?
Yeah, so we're building them, and we believe that getting the physical therapists early on is actually not a problem. We have a list of physical therapists online from the APTA you can find relatively easy. You can even find those who give telemedicine or allowing telemedicine tools online. And then every physical therapist we've talked to has only half of his workload or less. So people are at this moment very inclined to onboard in this type of solution.
I see. Thank you.
And any other questions?
Okay.
Yeah, Lynn.
Yeah, Matthias, really like ortho as a space. I think there's a lot of waste in the system because of this. And I would assume, PT, if you really go early in the consumer kind of clinical journey, if you will, there's a lot of cost savings that can come to that. Have you thought about kind of how you think about CAC and where you can kind of penetrate into the consumer patient journey earlier in the chain?
So yeah, we've been hearing about patients who've had injury, and that's a lot of sports people who don't really right away go to a physical therapist. We don't know exactly how to market to them, so that's something we want to explore. And the first thing we're going to explore is the online, to see if people look up for their symptoms, see if we can get them on board for a short introduction with a physical therapist.
Great. I think we'll stop there, and thank you, Matthias, and thank you, team. And let's call Chemical to the stage, Alex Pedron. So just some background on Chemical and Alex. Alex is a postdoc in biochemistry and biophysics. He is not on the fence. He definitely wants to do a startup. He's made that clear. And this is what he said about class. Class has been a phenomenal introduction to startups. I took this class because I am passionate about invention, about creation. I want to utilize my skill set to build something larger than myself and give back to the world because it's given so much to me. This class has provided me with the tools and blueprint to build and to create. I am very grateful for the experience, for having such a great instructor who has put together an excellent course. The team has been phenomenal as well. They are smart and hardworking individuals who feel similarly about startups. Alex, I just got to say, this is why we do this. Your motivation is why we're all here. We're not building apps for social media. We're doing something that matters, and I love your passion. So
appreciate that, Stephanie. Thank you.
Yeah, tell us about Chemical.
Okay. Can everyone see my screen?
Yep.
Okay, great. Okay, so thanks again, Stephanie. I'm Alex Pedron, and today I'm going to tell you about our company Chemical, a cell therapy platform focused on engineering safer cell therapies to treat cancer. I want to start off with an analogy. Imagine you're in a room filled with candles and you'd like to light a single one. The only problem is that what you have at hand is a flamethrower. How do you set fire to a single candle wick without lighting the others, let alone setting fire to the entire room? This is the problem current CAR T cell therapies face. They're very good at killing cancer cells, but they don't have control mechanisms to dial down the amount of fire being used or mechanisms to realize that the entire house is burning down and they should stop working. I'll outline the current safety hurdles for CAR T cell therapies that current CAR T cell therapies face in the coming slides. So first, CAR T cells work well for certain types of cancers, such as blood cancers, but no CAR T to date has been clinically realized for, say, solid tumors. And because of the way CAR T cells work, they can't localize to disease organs. They're specific to cancer. Second, CAR T cells have the interesting property of functioning too well, in essence. When a CAR T cell detects cancer and elicits a killing program, what often happens is that neighboring healthy cells get damaged in the process. This results in cytokine release syndrome, or CRS. About 30% of CAR T treated patients suffer from CRS, which can result in life-threatening reactions. CRS itself occurs because of a lack of control over existing approved CAR T cell therapies and amounts to about $250 million in total peak costs. CRS occurs because, in essence, CAR T cells get stuck in a positive feedback loop. And this happens because the cytokines used to kill cancer cells also kill non-cancerous neighboring cells. So the more, in essence, basically what happens is that the more cancer killing you have, The more cell death, and the more cell death, the more cytokine release, and the more damage occurs to healthy tissues until ultimately the patient manifests CRS. We are developing engineered cells that can sense when neighboring cells are damaged and automatically modulate the killing program they elicit in order to mitigate the damage of neighboring cells and ultimately of CRS itself. We believe our approach will result in effective cancer killing while mitigating both cytokine release syndrome and the additional $250 million total peak cost to treat CRS. Our overarching vision is to develop a cell therapy platform with better control, the ability to localize therapeutic cells inside the body, and the ability to target various indications beyond blood cancers. Here's an example of our engineered cells effectively killing cancer cells inside a living organism, shown here on the right. I don't have time to tell you about our in vivo tissue-specific targeting today, but if you're interested, talk to us later. The CAR T market is rapidly growing with a CAGR of 34% in 2019. We strongly believe our platform technology positions us well to target oncology utilizing our engineered cells. And in the future, we'll move beyond cancer to target other tissue-specific disorders such as inflammatory disorders. For now, though, our first product will focus on oncology. We have UCSF IP filed for critical components of our platform. And I've mentioned to you the downsides of CAR T, but CAR T has been incredibly successful in the market with several large acquisitions. A lingering problem remains though, and that's the cell toxicity problem. Our company is well poised in the competitive landscape with a platform that will offer safer cell therapies while mitigating adverse side effects. We plan on licensing our technology for clinical development with companies with proven manufacturing capabilities and clinical development capabilities. And we are also taking steps to de-risk our technology and prove it works robustly as a cancer therapy. It'll take $1.3 million to reach a value inflection point, but some of these costs will be mitigated by working in the lab at UCSF. By the end of 2021, we will have generated an optimized cell to test in mouse. Beyond that, we'll perform mouse studies for POC toxicity and additional cancer killing experiments. Our team is composed of 2 synthetic biologists and 2 molecular biologists from UCSF, as well as a pharma veteran. Our advisors are Hana El Samad, a UCSF professor, and Stephanie Maras, who of course needs no introduction. There are lots of opportunities in the cell therapy space, and chemical is a platform technology that's addressing cell therapy toxicity. Our first indication will be in oncology, and I didn't have time to go into detail on other important aspects of our platform, but if you're interested to learn more, talk to me later or email me at this address down below. Thank you.
Thank you. Excellent. Judges.
Thank you, Alex.
Sorry, go ahead.
After you.
Go ahead. I've been talking a lot.
Sorry, no, thanks, Alex. Actually, CAR T is very near and dear to us at Novartis. We launched Kymriah, one of the first CAR Ts in pediatric leukemia. Can you talk a little bit about, you mentioned a little bit about your business model regarding partnership. Is this an add-on type business model that is an additive to existing CAR T therapies? And kind of how do you think about actually forming these partnerships, and what does your kind of long-term business model look like?
Yeah, that's a great question. So I think we're really working on the next generation of CAR T cell therapies, although I do see an avenue for adding on added functionality to the current platform and gold standard. And in terms of our business model, you know, we are wanting to like I mentioned, seek partnerships with major cell therapy companies, but we're also considering fundraising for additional preclinical experiments to enable IND filing. And so that is still on the table for sure. And we're hoping to pursue both of those simultaneously.
Great. Alan, were you trying to say something?
I was gonna ask a question, which is really two quick ones. One is, can you just mention the tumor model that you showed proof of concept in, and then secondly, there are a lot of companies out there in CAR T and iBite that are using titration as a mechanism to various forms of titration as a mechanism to reduce CRS. How do you differentiate from those?
Yeah, so we differentiate pretty substantially, actually. Our rather than like so in other words, like if you set titrations up, the amount of time, like the feedback time is significantly longer. Our platform dynamically adjusts its killing program, and so a physician wouldn't even need to have like the sort of manifestations of CRS itself to know whether or not to dial down or up their the treatment. Our cells are doing it sort of autonomously, if you will. And in terms of the tumor model, this is I should say we're very early, and this is an engrafted K562 based tumor in mice.
Thanks.
Okay, we need to stop right here. Thank you very much, Alex. And I'm going to see if Pam is ready to present, or who is presenting for Pam. Let me know if you're ready.
Hi, I am here. I am trying to get the settings to work. I have spoken to Kyle.
Okay, we'll come back to you, Pam. We'll keep going. Okay, Isla.
I don't believe this.
Sorry.
Stephanie, can you at least give me the 10 seconds that's required for the video to pop up? I spoke to Kyle and he said it just needs to you need to just give me 10 seconds here.
You've got 10 seconds.
All right, Kyle, can you help me out? There we go. There's the share screen button. Thank you. All right, hello everyone. My name is Pam England, and I'm one of the founders of Neur1 Therapeutics. My company is focused on reinventing how CNS disorders are treated. One of the diseases that we're focused on is Parkinson's disease. Our goal is to develop a first-in-class disease-modifying agent that slows the progression of Parkinson's disease and results in superior clinical benefit and improved quality of life for patients. In a minute, I'm going to show you this video of Michael J. Fox, who was diagnosed with early onset Parkinson's disease about 20 years ago. But let me start by reminding you about what Parkinson's is. It's a progressive disease. It gets worse over time. There's no cure for it right now. It's the result of not having enough dopamine in the brain. And it manifests in three classical ways. One of those ways is motor symptoms. So patients with Parkinson's disease have stiff muscles. Sometimes they have a jitter in their extremities. Generally, they have depression because dopamine is part of maintaining a healthy mood, and their motivation is affected. These are all things that dopamine plays a role in the brain. I'm going to show you this video now, which will show you both the symptoms of the disease. You'll see that he has trouble speaking, and the side effects of one of the drugs that he's taking, L-Dopa. And that side effect will appear as jerky motions that he makes. So we're looking at both disease symptoms and side effects of the drug.
I wish I wasn't in this situation, but it's been one of the great gifts of my life that I've been in the position to take my view of the suckitude of it and merge it with other people's view of the suckitude of it and try to find an answer.
So he's talking about the suckitude of the disease and the drugs that he's taking to modify the symptoms. Again, all of the drugs available to treat Parkinson's are symptom modifying only. They have no effect on disease progression. And the gold standard drug, L-Dopa, which is a biosynthetic precursor for dopamine, loses efficacy over time. So there's a desperate need for new drugs. Our solution is to promote the endogenous production and release of dopamine. So rather than giving patients L-DOPA, which is again the precursor for dopamine, or drugs that inhibit the breakdown of dopamine, or drugs that mimic dopamine, what we're going to do is we're going to target a transcription factor, which is called NER1. And this protein, NER1, what it does when it's activated is it sits down on DNA and it'll drive the transcription of the genes that are responsible for making dopamine and releasing dopamine. So by upregulating those genes with our drug, we hope to correct the underlying imbalance. All of the pieces needed to develop these drugs are already up and running. And I'm showing you what they are on this slide. First, we have binding assays that allow us to measure the affinity of our compounds for the target. That's very important for being able to tune up how the drugs work. We have pictures, crystal structures for the aficionados in the audience, but they're basically pictures of the drugs bound to the target. Those pictures are an integral part of rationally improving the activity of our drugs. And then, of course, we have the gene transcription assays that allow us to determine what effect our drugs are having on the genes. And in the graph I'm showing you on the right, one of our compounds you see upregulates the synthesis gene by 150% and the release gene for dopamine by about 200%. So we have all of this working already in the company. The market opportunity for Parkinson's is really large. All of the drugs right now, which I've listed them at the bottom of the slide, are valued at about 3.7 billion annually with a 6% growth every year. The add-on drugs, which are all the drugs except L-DOPA, the gold standard, constitute about 1.5 billion a year. And then the only proprietary drug currently on the market has a market value of about 0.3 billion per year. Depending on how we enter, we may be competing with the proprietary drug, but eventually we hope to be competing with all drugs because all the drugs are symptom modifying and ours is going to be disease modifying. We have minimal competition in the space right now. Although there are a lot of companies working on Parkinson's disease, very few of them are working on disease progression. There are half a dozen companies that I'm showing on this slide on the left that are in phase 2 clinical trials or beyond, but they're focused on symptom management. And then there are a handful of companies I'm showing on the right that are focused on disease progression, but 2 of those companies are focused on the genetic forms of PD, and that only comprises about 10% of the patient base. There's no one in clinical trials right now that's targeting our target NER1. Our intellectual property surrounding this entity is good. We have 2 patents, one non-provisional, which has 2 lead series compounds in it. They're composition of matter patents. And we have a provisional patent that we'll be filing this month. Our product development over the next 12 to 18 months is shown on this slide. The first year is going to be spent finishing up the discovery phase of the project, but we hope to reach a key inflection point by about May of next year. And that inflection point is completing efficacy studies in PD, Parkinson's disease animal models. And by 18 months, we hope to be in phase 1 clinical trials. Our NER1 Therapeutics team is comprised of myself and Matt Jacobson as co-founders. I've raised significant non-dilutive funding for the project, including from the Michael J. Fox Foundation. And my co-founder, Matt Jacobson, has started and exited from several companies already in his career. In addition, there are a handful of really terrific advisors that have been helping with the project: Pat Sinatra, David Morris, and Peter Neumer. There are more on the business side of things, but have been very helpful. And then my classmates on the team, Clay and Luca, have been really helpful doing the interviews that we did to get our information and putting the deck together. In summary, we're targeting a large underserved market. We have a different biological target and a novel mechanism of action compared to everybody else out there. We have minimal competition in the field, in the space. We're building very strong patent estate, including composition of matter patents, and we have a team with successful startup experience. Thanks for your attention, and I'm happy to take any questions.
Okay, judges.
So obviously, the big question, I guess, is why is no one else going after NER1? I mean, everyone's going after some, fetish targets like alpha-synuclein. And why is that? Obviously, the L-DOPA mechanism has been known for a while.
Yeah. Yeah. So there's a really long answer to that, but the short answer is that in the mid 2000s, there were a number of big pharmaceutical companies, Amgen, Novartis, Pfizer, that did try to target Nurr1. And there's a little bit of a disconnect in the science between binding and activity. And then all these pharma companies gave up their CNS programs. So we have actually solved the disconnect between binding and activity. I pointed out we had 3 different binding sites. We know which binding sites lead to activity and which ones don't. Another hurdle is the first crystal structure of Nurr1 was published in 2003. There was no ligand bound to it. 15 years later, my lab published the only other crystal structure with a ligand bound. That ligand happens to be an endogenous metabolite of dopamine, and that paper's been published. But we have additional crystal structures with synthetic ligands bound that we haven't published yet. So I think the disconnect between binding and activity and then no crystal structures just kind of put a kibosh on Nurr1. But the Michael J. Fox Foundation listed it as one of their top 5 priority targets about 5 years ago. So it's back in the limelight, and I think people will be going after it, but hopefully we have a jump.
Great, thank you.
Yeah.
Okay, I think we will stop it here. Thank you, Pam.
Thanks.
And yeah, the next team is Isla and Rory Stanton. So while Rory comes to the stage, she's an MBA student at Haas, and she's teamed up with a UCSF bioinformaticist, Charlotte Nelson, who's in Atul Butte's lab. I'm sure many of you know who Atul is. So this is what Rory had to say about her reasons for taking this. She said, I'm graduating from Haas this May, and my goal was to launch a startup out of grad school. I came to UCSF with a problem that needed to be solved and was looking for a scientist who could help me do it. I found my co-founder through this class. Startup 101 has widened our perspective and introduced us to concepts that will enable us to transform the healthcare industry. So Rory, please share your screen so we can see your slides. Great. Are you ready to go? Unmuted?
Yeah, let me just unmute.
You're unmuted. You're good.
Oh, I'm unmuted. Awesome. Hi, everyone. My name is Rory Stanton, and we are Isla Health. We are a precision health platform for complex chronic conditions. This is Shannon, my cousin. About 7.5 years ago, she was living in London, had just graduated from a top-tier MBA, and from the outside seemed to be on top of the world. But what we didn't see on the inside was that things were slowly crumbling. She started to develop very odd symptoms that she couldn't quite make sense of, and thus began her journey over the next 3.5 years, going from doctor to specialist, and back to the regular doctor again, taking innumerable tests, only to find out 3.5, 4 years later that she had an autoimmune condition. I started to think about why was it taking so long for her to get an answer from what she was experiencing. So I started to dig into this problem a little bit last year. At the end of last year, I began the NSF Core program, and since then I've conducted over 70 healthcare stakeholder interviews across patients, health systems, providers, you name it. And we started to hear this story ring true over and over again. Patients would say it took 5, 10, sometimes 15 years for a diagnosis. And providers similarly commented that because of the vague nature of these symptoms, it's extremely difficult to diagnose an autoimmune condition. And some startling facts you may not know is that 1 in 6 Americans is living with an autoimmune disease today, and fully 24 million of those are not yet diagnosed. We believe there needs to be a change to this problem, because delays in diagnosis and treatment accelerate costs rapidly for health plans. It can be up to 25 times higher in per member per year spend for an autoimmune patient compared to the average. And while this is a huge problem for value-based care systems, we believe there's a huge opportunity for Isla Health. About $18 billion is spent in autoimmune care waste every single year on those unnecessary visits and unnecessary tests due to the fact that patients aren't routed to the right care provider at the right time. So we developed a solution to change that. We have an application in which patients can track their symptoms and quantify progression of those vague conditions and symptoms remotely. But we don't just track. We actually map this patient-generated data into our AI knowledge engine, which is powered by thousands of biomedical research studies and multi-omics data. When that AI is able to help us discover the underpinnings of these diseases and how they're manifesting in each and every patient, which enables us ultimately to do better patient stratification and earlier detection in the journey. We utilize the AI in order to route care according to the patient's underlying biology. So whether it means wait and watch and do a remote monitoring program, direct them to our network of telehealth providers, send them into the right specialist at the right time, or even get them to emergency care when they need it. Moreover, on the provider side, this data syncs automatically so that they are able to generate the right insights about the right patient in the right time. We believe that we'll have rapid adoption of this technology in the marketplace due to expanded coverage under new CPT codes with remote monitoring, telehealth visits, and chronic condition management as a result of changes in the 21st Century Cares Act. We have a pretty clear regulatory path as decision support software that's solely working on providing insights is not yet regulated by the FDA. In the care navigation market, there are other players that are basing their algorithms on clinical guidelines, whereas ours takes a look at the underlying biology of a patient and routes them appropriately. Our business model is three-tiered. We're marketing to telemedicine providers a network in which they can participate and generate referrals through our platform. We're also partnering with health systems to provide patient management software on an annual licensing fee basis. And downstream, we'll work with pharmas for clinical trial recruitment, as well as conducting real-world evidence studies for therapeutic discovery. We plan to have a cash-efficient distribution model by partnering with payers, EHR apps marketplaces, and diagnostics companies. All that said, we're in the middle of a crisis, and we felt because our application has the possibility and potential to expand to many different kinds of diseases, we're going to start focusing our efforts on COVID-19 today. And as a matter of fact, over the last week and a half, actually 8 days probably, we've rapid prototyped our new solution and plan to launch it early next week. This COVID-19 solution will help patients manage their care remotely, reduce inbound pressure on frontline healthcare resources, and help scientists get the data they desperately need to discover new insights and help inform new therapeutics. Our roadmap for the next 12 months is to launch this into the direct-to-consumer marketplace, get a critical mass of patient signups, partner with health systems to get this application out there so that they can better manage their patients from a distance as they're self-managing their care in their homes, and ultimately be on the front lines helping healthcare heroes today. In summary, we're going to market with a COVID-19 solution. Our AI accesses thousands of research studies to understand the underlying patient biology, and we're looking to reduce the time to diagnosis for autoimmune patients like my cousin Shannon. Our team is led by business professionals such as myself and a team of wonderful, brilliant scientists who have been working on COVID-19 since we started in January. And we also have a wonderful network of advisors who are experts across the healthcare industry and serial entrepreneurs. We are interested in partnering with anyone who's interested in implementing our COVID-19 solution. You can contact me at this, and please stay safe and healthy. Thanks so much.
Thank you, Rory. And here's an example of a team of entrepreneurs who jumped on this situation, saw an opportunity to utilize their technology for COVID, and stayed up for how many nights in a row, did you say, Rory? 4 or 5?
We're still going.
Still doing it, still staying
like
up.
7.5, 8. That's why I look so tired, but
oh my
yeah,
gosh.
together.
Okay, well, congrats for doing that. Judges, who wants to kick it off?
Have you built the database using the 1,000 research papers for autoimmune disease, or are you about to do that?
It's actually applicable to many different diseases. We're starting with autoimmune because that was the kind of use case that we found really interesting that we wanted to go after, but it can be applied. It's a platform technology, so it can be applied to a number of different kinds of conditions.
But have you actually built the database yet?
Yeah, we have. Yeah. It's come out of research from my co-founder, Charlotte.
Great, thank you.
Okay, who else judges? Neil, Lynn?
I'll jump in here. You know, I've seen versions of these, you know, very thorough assay for chronic care management platforms. I think Driver was probably one of the more extensive ones in oncology space. You know, I've seen them. The barrier to entry is relatively low. When you start adding in sort of the biological interpretation, it gets, or the actual assays themselves, it gets slightly higher. And I'm wondering, you know, with respect to how you want to differentiate yourself and go to market, where are you in comparison to kind of like several of the others who have kind of failed here?
I think our goal is to utilize kind of the swift market entry by doing the DTC low regulatory. And then as we develop our kind of proprietary knowledge and data, we're going to look at expanding into other more difficult areas and potentially looking to SAMDs.
Okay, I think we're out of time for questions. Thank you very much.
Thanks.
Isla Health. And our next up is Previ, Sarah Fernandez. Sarah, if you want to come to the stage. Sarah is a Haas grad and a PhD who describes herself as a business leader with more than 10 years of experience in healthcare business development. She's been working on her startup for over a year and took this course to connect with UCSF and explore a new market. So her comment was, it was a great working team and she's established relationships that will continue beyond the class. So that's always good to hear. And Sarah, are you there? Sarah?
Hi, I'm here.
Yes, I can see you. You want to share your slides?
Great.
Okay, we can. Great.
Can you see my slide now?
I can, yes.
Great. So thank you, everyone. My name is Sara Fernandez, and I'm a co-founder and team lead of Previ, where we are expanding patient care to outside the clinic. The past few weeks have really underscored the need for a digital transformation of healthcare. Most providers are delaying non-urgent care, or they are scrambling to contact their patients at home. It has really underscored the need for systems that connect to the patients when they are at home. The reality is that outside this COVID-19 crisis, most of the care still happens the same as 30 years ago, inside the four walls of the clinic and limited to those 15-minute interactions between the doctor and the patient, when the patients spend most of the time outside the clinic. Take this patient, for example. She's alone at home and she has a lot of questions about her treatment, about the side effects, and then she has trouble coordinating all her healthcare data between her local primary care physician, her oncologist, her radiologist, and the diagnostic labs. This is almost like a full-time job. And in fact, wealthy patients many times hire nurse coordinators that guide them through that process. Now we want to bring Previ to support the majority of the patients. On the other side, the provider has no view on what is happening to the patient when they are at home. Previ supports the providers in this digital transformation. We start with providing a venue for coordination of care. Instead of being tied to the current fragmented care that is institution-centric, Previ moves to a more patient-centered care where all the members of a care team can share patient information and care plans. For the patient have guidance through the journey when they are at home. They can also have a HIPAA-compliant communication with their doctors via both video conferencing as well as chat. And then finally, the providers have visibility to what is happening to the patients, both via remote patient monitoring of medical devices, as well as through patient assessments. We started the company targeting independent primary care physicians for two reasons. One, there was a clear reimbursement from Medicare to promote this type of remote care in primary care. And two, independent physicians have a shorter sales cycle than larger customers. But now we are moving to larger customers. We are targeting both health systems and ACOs, where coordination of care is a critical component and is still a problem unsolved. And then we are also having conversations with pharma companies that want to offer, want to go what they call beyond the pill and offer patient support services that guide them through their treatment and at the same time want to collect patient-reported outcomes. During this class, we wanted to explore the cancer care. And when you see in our team, we had a lot of cancer specialists which made this journey much easier. Medicare is starting to penalize hospitals for visits to the emergency department or hospitalizations for patients related to chemotherapy side effects that can be prevented. And we have estimated that within this market, we could target the 900 million market for care that is reimbursed by Medicare to provide this remote care. We help providers identifying high-risk patients based on clinical decision support. We help them with the coordination of care. And then finally, we help them standardize symptom management and at the same time guide the patient through that symptom management. So we can target the problems when they are in the early stage and not have to wait when they are in a crisis. This is the view for the provider. We can customize the dashboard that we show to them depending on whatever metrics they want to show for their patients and create alerts based on using clinical decision support tools and based on data that they are receiving directly from the patients. We are in the process of integrating with major EMRs. We'll complete that in the next 3 to 6 months. For the patients have an end-to-end solution to guide them through their treatment. And these digital care plans adapt to each patient situation based on both their clinical history as well as to the data that they enter into the system. So a digital care plan can include medication reminders, patient education, remote monitoring, patient assessment to collect these patient-reported outcomes, and then appointment reminders. We are currently live with some early customers. We have 6 doctors in 2 clinics in Miami, and we have about 2,000 patients enrolled. We have seen that the retention rate is 13% after 8 months, which is comparable with other solutions in the market. We were recognized by the Office of National Coordinator as in their Consumer Health Data Aggregator Challenge, and then also by Novartis in a recent bioderma competition in Barcelona, Spain. There are many other solutions in the market. What we have found is that most of them, they either focus on a single or 1 or 2 services that they apply to multiple conditions, like MediSafe, Mango Health, or they focus on a complete solution that they are using for 1 or 2 diseases, like GlucoCare, Omada. And those larger companies that everyone recognizes, most of the time they develop their own protocols, and they give little flexibility in adapting them to its institution. With Previ, we provide the flexibility of using some of our already developed care plans, or use our clinical decision support editor to create your own plan or customize the ones that already exist.
You have 10 seconds left, Sarah.
Okay, this is the Previ team, very heavy on clinical and oncology expertise. And now our next months, we are going to be focusing on sales. Like Mark Cuban says, sales cures all. So that's going to be our main focus.
Great.
I hope
Okay.
that introduce
Judges.
and convince you that Previ is a great end-to-end solution for remote patient care. Thank you for your attention.
Okay. Lynn, do you want to kick this one off?
Yeah, sure. Sarah, thank you. I think it's great to see that you're focused in one area like oncology. I think it tends to be in a lot of these platforms that you go after multiple conditions. So I think focus is really important. Can you talk about, it sounds like physicians are your main customer, kind of what types of physicians are you going after? And then what's the value drivers for these physicians to pay for your solution? And kind of what they see, are they in value-based care arrangements? Just talk a bit about that.
So we have two different business models. For providers that are in fee-for-service, like I mentioned before, now Medicare is starting to penalize them for preventable hospitalizations. So they have an incentive to have this type of solution. And at the same time, there's a new CPT code that pays the providers to offer this type of solution. So they have a good reason to use our services, increase their revenue, and at the same time avoid hospitalizations. For providers in value-based contracts, again, this is a very efficient tool to reduce costs that can be prevented in chemotherapy patients.
Only a few seconds left for Neil to jump in. I think he wants to. Go ahead.
Yeah, just a quick comment on looking at your competitive landscape. It's pretty broad and it focuses on a lot of point solutions. I think there are other clinical decision support solutions you might want to take a look at, and we can list them separately. My specific questions on scalability. What's your plan for driving scalability given how fragmented the market is?
So we are looking to partner with channel distributors. One is clearly the EMR marketplaces, and then we are also talking to technical consulting companies that many times do these digital transformation processes in large healthcare institutions.
Okay, we need to move on. Thank you, Sarah. And hydrocephalus, Sung Hoon, come to the stage. We are down to the last two teams, so thank you out there for everyone who's stuck with us. We've got a medical device team and then a digital health team coming up. So Sung Hoon, are you here? You are. I can see you've shared your screen. And so Sung Hoon represents a team of UCSF pediatric surgeons and a biomechanical engineer who are working on a devastating problem. And Sung Hoon said, I am taking Startup 101 because I want to make sure that the pressure sensor we developed gets used for patient care in the future. So please, Sung Hoon, go ahead and tell us
Thanks very much.
What you got.
Okay. I don't know what happened. It just disappeared.
We're good. I
can't see anything.
Oh, I can only see there's a diver swimming. So I guess your slides are disappeared. There they are. You got them.
Okay. Thanks very much. My name is Sung-Hoon Kim. I represent Team Hydrocephalus Technology. I'll be talking about a medical device, implantable intracranial pressure sensor. This is a picture of an infant who has hydrocephalus who was untreated. When excessive fluid accumulates within the brain, hydrocephalus results. Hydrocephalus is not curable, but it can be treated with a shunt catheter. A shunt catheter diverts the fluid from the brain into the abdomen. Unfortunately, the shunt technology is not perfect, and shunt often becomes clogged. Up to 50% of shunts are replaced within 5 years due to obstruction. Diagnosing shunt blockage is not easy. It involves radiologic studies such as MRI and surgical exploration. Berkeley is a patient with hydrocephalus. Due to shunt malfunction during her lifetime, she had undergone 30 shunt operations. Her headache returned after each operation. She had to endure many surgical procedures to find out if her shunt was blocked or infected. We need a non-invasive, simple way to diagnose shunt obstruction. Our solution is an accurate, wireless, batteryless pressure sensor. The prototype is shown on the left panel. The sensor is placed in line within the shunt with an external reader which powers the sensor. The shunt pressure data can be obtained and displayed on a cell phone. Our value proposition for our technology it simplifies evaluation of shunt failure, reduces medical costs in evaluating shunt malfunction, it prevents unnecessary operations. Shunt catheter market in the United States is $300 million. Hydrocephalus treatment market is $1 billion in the US. The world market is $2 billion. There is no FDA approved device for wireless shunt pressure sensor. There are research projects in progress, for example, at Northwestern University and in Germany. Our competitive advantages are the sensor is connected to the shunt catheter. It is extremely reliable. It has been underwater tested at 37 Celsius continuously for the past 4 years. The device has no measurement drift. To protect our intellectual property, a US patent was obtained in 2016. Additional provisional patent was filed in 2019. We intend to seek Class 2 classification and pursue 510K pathway using predicate devices such as CardioMEM, which is a cardiac pressure sensor. Once FDA approved, we intend to sell the device to hospitals as a distributor. Our exit strategy is to license the device to shunt manufacturer. Reimbursement strategy is to bundle the sensor with existing shunt catheter systems. There are existing CPT codes that can be utilized. We plan to obtain $4 million Department of Defense grant. This is based on our confidence in the reliability of the prototype device that we have made and the existing need that DoD has announced for US veterans. Once funded, we'll begin animal testing next year. With the research data, we'll seek 510K clearance in 2023. The team consists of pediatric surgery, neurosurgery faculties from UCSF, and engineering faculty from Caltech. Advisors are Stephanie Marous and Howard Edelman. In summary, shunts often fail, and there is no easy way to determine shunt malfunction. Our pressure sensor will become the standard of care for non-invasive shunt evaluation. Thank you.
Thank you, Sung-Hoon. We finally have one for Alan May, who's a device guy. Go for it, Alan.
Yeah, I was wondering if there was going to be anything for me there.
Sorry, we didn't have a lot of devices this year.
No, that's great. It's been a very interesting evening. I wasn't very clear though on the extent to which you're invasive or non-invasive. Are you touching the fluid, or are you accessing at all the interior of the catheter, or are you reading externally through the catheter walls?
The sensor is incorporated into the existing DP shunt. So it's connected to the shunt itself. So it reads the pressure inside the shunt catheter.
Thanks for this. So if the shunt manufacturers decide that that they don't want to do that, or they can do that without you, how do you proceed?
They are already doing it. They are using the shunt without any sensor. If we introduce a sensor, now you have the means to detect when the shunt has failed. This is something that we really need to have.
Okay. Thank you, Hydrocephalus and Sung-Hoon.
Thank you very much.
We are ready for our last team. Catherine Grill, come on up. Neolith. Catherine is a behavioral neuroscientist who specializes in integrative and behavioral medicine. She launched Neolith and wanted to experience Startup 101 to hone her business plan. I told her, maybe you're too far advanced for this class. And she said, no, I want to take it. And she's been a faithful participant. This is what she had to say about class. The support we received during Startup 101 has helped us with lead generation, scaling our evidence-based sales model, and helped us win the TechStars Award at the SoGal Global Finals, largest global pitch competition for female founders. The mentorship we received was invaluable. So Catherine, please tell us about Neolith.
Hi, Stephanie. Thanks so much for having me. Can you hear me okay?
Yeah, we sure
Okay.
can.
And you can you see my slides?
Yes.
Okay,
You're in good
great.
shape.
Great. Oh, hold on. Sorry, it's restart these. I think I've lost screen sharing. Hold on, let me bring it back up. Thanks so much, everybody, for being here till the end. I appreciate it. I'm very excited to share our company, and thanks, Stephanie, for putting on this great event. All right, are you able to see now?
No, can't see your slides.
Okay. Oops, sorry.
You had them and you lost them.
I did have them and I lost them. Share screen.
So while Catherine's doing that, here it is. Well, I'll just tell everybody in the audience that the judges will be meeting separately after this session, and we will decide who the top 2 teams are for the prizes I announced earlier. And I want to thank the judges for their participation tonight. Catherine, go ahead.
Great. So hi, I am Catherine from Neoth, and we are using precision neuroscience to simplify behavioral treatment matching. I want to tell you about Alex. She's recently started college and is feeling pressure to succeed. If her stress is left untreated, it could lead to mental health problems. She may drop out. She could even be like 20% of her classmates and consider suicide. Universities are desperate to help students like Alex, which is why they dedicate billions of dollars per year to mental health services. They're looking for innovative ways to deploy these funds because current solutions fall short. Alex went to her campus counseling center to get help managing her stress, but was told she needed to wait 60 days to get an appointment. But she needs help today. So she talked to her friends about finding a digital program, but they realized that they all react to stress differently. As a neuroscientist, I know this is due to neurodiversity, individual differences in their brains. This means that they all require different treatments. So for Alex, a worrier, she does well with mindfulness, but her friend Jamie benefits from journaling. So how can they find a program that meets their neurodiverse needs? The answer is with Neoth. We used brain imaging data from thousands of patients to identify brain biomarkers of stress and which treatments were best for each biomarker. We call it precision neuroscience, and we're the first solution on the market taking this approach. Here's how it works. Our neuropsychological screening tool analyzes stress behavior to identify weakened brain areas. Then our algorithm curates evidence-based behavioral practices to strengthen those specific brain areas. These practices are updated over time based on progress. Here's what Alex experiences. She completes our screening tool. It's a survey that takes about 10 minutes. We ask her questions about her stress and behavior. Our algorithm takes this information to build her a personalized stress management plan. She gets practices like breathwork, guided imagery, or art therapy that she can do on our platform. Our algorithm uses Alex's feedback and machine learning to update these practices. And each month, Alex gets a progress report that she can share with her school counselor. Alex loves Neoth, and she's not the only one. We have a solution on the market, and this is feedback from a real user. It's our goal to help 1 million students like this over the next 5 years. Since the campus closures, we've seen increased student signups and an immediate need for our solution, helping the students manage stress and for the universities to collect data analytics on student emotional and physical health, as well as our new feature of financial health that we've just added. Neoth is the only solution using precision neuroscience. Our platform works with 40 diagnoses and 7 treatment techniques, so we're generating unique data and have many clinical applications. Our Startup 101 team is passionate about mental health. As a professor, I taught precision neuroscience to future therapists. Kelly founded a student mental health organization in partnership with NAMI, and Naveen is a therapist specializing in stress management and addiction. We have the team to scale this company. My co-founder is an engineer and IT project manager. He managed multi-million-dollar projects, including the software development for the Golden Gate Bridge tolling system. Florian is a full-stack developer with 15 years of experience, whose previous company was sold to Siemens. Dr. Wheeler is a renowned expert in stress with 20 years experience in student mental health, and we have Stefan in sales to close deals. Our advisors are seasoned entrepreneurs and domain experts. We're targeting the 40,000 universities and private high schools in the US. They charge health fees each year as part of tuition. 40% of this is spent on mental health, and that number is increasing. With our current pricing, we can make over $1 billion. But since everyone experiences stress, we could expand to other markets in the future. We work directly with universities who pay an upfront annual price per student so their students can access our platform. A recent sales campaign shows we have a 10% chance of moving to contract negotiations. Initial data indicates that our sales are improving with the campus closures. We're scaling up our evidence-based sales model over the next year by contacting 10,000 university executives and tracking our progress. We're so proud of what we've done since September. We have a product on the market and we're generating revenue. We're part of Startup Health and AWS EdStart. Our university partner already asked for a second year with us, supporting thousands of students and employees. We recently won the TechStars Award at the SOGA Global Pitch Competition from over 1,700 companies. With this award, we have the opportunity to join the TechStars network, receive funding, and roll out with their university partners across the country. We're adding more schools to the pipeline every day. In the next weeks, we have an invited lecture that we're giving at Carnegie Mellon and a paid workshop at Amazon. We also have research underway with leading partners. With $500,000, we can help 100,000 students manage their stress over the next 18 months, rolling out in 25 universities by the end of 2021. With Neos, students like Alex don't need to wait to get access to care. Her and all of her friends can get care that's personalized for their neurodiverse needs. And universities can provide 24/7 support that's cheaper than keeping the counseling center open around the clock. I'm Catherine from Neos, and we are using precision neuroscience to simplify behavioral treatment matching. Thank you.
Thank you, Catherine. Well done. Judges, who wants to start?
Yeah, Catherine, this is Lynn. Oh, go ahead, Richard.
Go ahead.
Then you're on.
Sure. So, Catherine, can you talk a little bit about, the, if you will, kind of the PRO tool or the kind of tool, the diagnostic tool you're launching, and then how it actually affects PHQ-9 or GAD-FLT7, or what type of outcomes you've had, I assume, maybe in developing these tools in the past. Just a little bit of the clinical data behind it.
Yeah, so with my background, I was at first working as a therapist and then switched over to neuroscience. And for the past 8 years, have really been looking into the neural mechanisms of behavioral therapies. So what's going on in the brain when they have all these different symptoms, and what's going on in the brain with all these different treatments. So the idea is, for the past years, I've been sorting through thousands of neuroimaging studies and aggregating that data. And what we did with that data is we built an algorithm that can pull that self-reported information about the patient's stress and behavior, pull that information, assess which areas of the brain are likely impaired, and which behavioral treatment would be the best appropriate treatment for that person. So it's a neuropsychological screening tool that we use.
Rich, did you have something?
Yes. Do you have a feedback loop where a student may need serious intervention? You know, and who would they contact in that case?
So it would depend on the university partner, and that's something that we would make specific to each partner. There is usually a counseling center on site and a director of the counseling center that they could contact. But because they're not open 24/7, a lot of universities will partner with maybe a hospital or another clinic in the area that is open 24/7. So it is client specific who they would need to contact.
Okay, thank you.
Great. Okay, well, let's call that a wrap. Thank you, Catherine and team.
Thanks so much, Stephanie.
So just for all the teams, what a great job you did tonight. I mean, can you believe, audience, that they learned all this in 12 weeks? They started with nothing, and with a couple of exceptions, like Catherine, who's been doing this for a while, but most of these teams had absolutely nothing, and look at all they've learned. I'm always just amazed at how incredible our students are. Students, I'm going to miss your intellectual curiosity. It's really been fun teaching you. And audience, I want to thank you for coming tonight. I hope you were given a good feel for what happens at UCSF behind our walls and how we're generating interest in entrepreneurship. Please sign up for our newsletter, stay in touch. And if anyone out there is interested in learning more about a team, get in touch with me. My email is stephanie.maris@ucsf.edu. For our entrepreneurs, go forth and win, make something happen. You guys are great and you can do it. I know you can. And I will tie off here and thank you so much for coming.
Here.
Thank you.
Thanks, everyone.
Thank you. Great job.
So are we dialing?